Hello everyone, Dr. Suresh here, and in this video I will be going over an important concept that has been repeatedly tested in Neetpg and Fmge. Let's have a look on the case that appearing on the screen. So here look for the key terms, key words, okay. Just I will be reading the question, just be with me. A 20-year-old overweight male, okay, you see here overweight male was into self-induced weight loss. What is a thing? Self-induced weight loss plan, and it has been more than 45 hours since he had last meal. 45 hours since he had last meal. These are the two things you have to keep in mind. Except he is on a plain water. His blood glucose levels are normal. Okay, what is another key thing here? Blood glucose levels are normal in range by the liver. One of the initial and primary enzymes involved in conversion of non-carbohydrate sources into glucose is located in hepatocyte mitochondrial matrix. Okay. So the question has been asked here is, which one of the following molecule increase the activity of this enzyme? So they have said that some mechanism is acting to supply glucose from the liver cells, and to start this pathway, what enzyme involved here? Okay, and that enzyme activity increases by which compound? There have been 4 options has been given. One is alanine, B acetyl CoA, NAD plus and lactate. Just have look. I am just giving you 1 minute time and be ready with your answer before the explanation. Okay, let us begin the explanation, the answer for this question. So before giving the explanation, I want to put 3 points here. One is, what is the metabolic process involved? What is the metabolic process involved or discussed in the case stem? And second is, important enzyme of the discussed metabolic pathway. Important enzyme of the pathway. Third thing to know, activator of the important enzyme. Activator of that important enzyme. So this is like reverse learning from going from bottom to top, right? So we have already come to know that what's happening here, like fasting condition. He is already on diet plan, that means he is not having anything, that means 48 hours, 2 days. So I have already seen in fasting conditions, if the person is not having anything, what are the backup mechanisms will come into the action. First thing is, if the person is not having anything, if his blood glucose levels are low, first what will happen? First thing to be happen, that is glycogen breakdown. Glycogen, okay, but whatever the amount of glycogen stored in liver and muscle will supply you up to 40 hours. But here our case, the person is without food more than 48 hours. But whatever the glycogen stores in his body has supplied the energy up to 40 hours. Then what to do above 40 hours? So there are other, glycogen is a carbohydrate, right? So our body can able to synthesize glucose from non-carbohydrate sources. Then the gluconeogenesis pathway. So here first our point, gluconeogenesis. Then how this gluconeogenesis will start? Gluconeogenesis will be when there is a glucose will be in the beginning state. After 12 hours of food, gluconeogenesis will start. 24 hours it will be peak. After 24 hours the peak will keep on going until you provide glucose, right? So now gluconeogenesis, how you say? Gluconeogenesis, how you define? Gluconeogenesis is nothing but synthesis of glucose from non-carbohydrates. Synthesis of glucose from non-carbohydrates. So what are the non-carbohydrates? I will show you here what are they. One is pyruvate, lactate, alanine and glycerol, propenyl CoA. So what are the sources of this? Pyruvate, anyhow it is coming from the glycolysis, glucose will be converted to pyruvate, that is end product. And lactate, pyruvate in anaerobic conditions, pyruvate will be converted to lactose and lactose can convert back to pyruvate by lactate dehydrogenase. Alanine, because we don't have any we don't have any source of glucose, so here proteins will broke down to amino acids. Amino acids, we have already learned the classification. What classification we have learned? Based on metabolic fate, glucogenic, ketogenic and both, right? Here glucogenic amino acid which involved in production of energy will be released. One such example is alanine. And glycerol, alanine is from protein, okay, remember alanine is from protein breakdown and glycerol is from fat degradation, okay. And propenyl-CoA, because of the breakdown of branched chain fatty acids and phytanic acid peroxisomes, okay, branched chain fatty acids and phytanic acid breakdown, they are releasing. So they all reach in circulation and they reach to the liver. So now we will see what is important enzyme involved in gluconeogenesis. Here to know about, first thing, pyruvate is a 3-carbon compound, will be converted to oxaloacetate which is a 4-carbon compound. Okay, pyruvate is a 3-carbon compound, oxaloacetate is a 4-carbon. How it is possible? Here the first enzyme of gluconeogenesis comes into the action, that is pyruvate carboxylase. Remember what is the enzyme? The enzyme is pyruvate carboxylase. Pyruvate carboxylase. Here 3-carbon pyruvate will be converted into 4-carbon oxaloacetate. That means carbon is been added. How it is added? It is adding the carbon CO2. CO2 is a donor. And what and all things it require? This carboxylase is what type of enzyme? It is a ligase. It belongs to the class 5 enzyme. It adds the carbon to it. It makes a bond, right? So obviously we learnt from the enzyme classification that ligases require energy, right? They are energy dependent. Here ATP involved converted to ADP, right? And it is also require magnesium, and and we all learned that when pyruvate enters into mitochondria, this will be converted to oxaloacetate. So here pyruvate carboxylase, what you come to know, it is a mitochondrial enzyme. Remember one more thing, it is a mitochondrial enzyme. Okay. So this is a thing, it is converting 3-carbon pyruvate to 4-carbon pyruvate carboxylase, and here the main activator, the question in the case asked, what is the activator? The activator is acetyl-CoA. Remember the activator for this enzyme is acetyl-CoA. In our body there are two reactions that exist. One is the acetyl-CoA acting as an activator. One is activation of pyruvate carboxylase and the other one is CPS1. But how CPS1? CPS1 involved in urea cycle. Okay, CPS1 involved in urea cycle where CO2 and ammonia combine to form carbamyl phosphate. The enzyme is carbamyl phosphate synthase 1 and the activator is N-acetyl glutamate, and for that N-acetyl glutamate formation acetyl-CoA acting as an activator. Okay. So N-acetyl glutamate is required, but for again N-acetyl thing donated by acetyl-CoA. Here that glutamate acetyl-CoA combined to form N-acetyl glutamate. For that acetyl-CoA is required. So there are two reactions in our body where acetyl-CoA acting as a positive modulator or activator. So the question now is clear for you. So what happened here? The question, which one of the following molecule increase the activity of the enzyme? Options already has been given. Now so far we have explained. So let me explain further and we will jump to the answer. So we have said pyruvate has been come, then one more thing here, lactate. Lactate can be converted back to pyruvate. What is the enzyme? Lactate dehydrogenase. The enzyme is lactate dehydrogenase. It is a reversible reaction, lactate to pyruvate, pyruvate to. And here oxaloacetate also can be converted from alanine. Okay, alanine coming from protein breakdown. Cortisol, when you don't have enough glucose amounts during fasting conditions, cortisol releases, and this cortisol induces protein degradation to form glucogenic amino acid to produce energy, and it will be converted into oxaloacetate, and otherwise this alanine again reaches to the liver and it will be converted into pyruvate, right. So, this is the mechanism how things work, and now further metabolism we will see. This oxaloacetate, this synthesized oxaloacetate will further be converted to malate, and this malate again converted to oxaloacetate. This oxaloacetate enters into cytoplasm, and this will be converted back to phosphoenol pyruvate. What is the enzyme here? Phosphoenol pyruvate? Phosphoenol pyruvate carboxykinase. This is one more enzyme in gluconeogenesis. The second important or key enzyme in gluconeogenesis, phosphoenol pyruvate carboxykinase. Okay, and here GTP is involved. It is GTP dependent enzyme. Here GTP is involved. Understand? So, now let us see the options. Now, from this phosphoenol pyruvate there are sequential steps to synthesize glucose. Okay, and this glucose is a contributor of blood glucose. Okay. So, these all pathway happening in the liver, and that is why liver is a contributor. If you see the thing here where blood glucose levels are normal in range by the liver, because gluconeogenesis is taking place in the liver. So, that is why glucose is synthesized and supplied to the blood. Now, let us see the options. What are the options are here? One is alanine, because we have already learned that alanine is a substrate, is not an activator. Okay, from alanine pyruvate will be converting, alanine is a substrate of gluconeogenesis. Then how come we are activator? No, it is a ruled out option. Next is acetyl-CoA. So, we have already clear cutly we have learnt that acetyl-CoA is the activator of pyruvate carboxylase. Okay, acetyl-CoA is the activator of pyruvate carboxylase. So, the option is acetyl-CoA, it is correct, right. And the third option NAD, no way. And lactate, yes, lactate, alanine both are substrate, how come they will be activators? I mean, no way. So, the correct answer is acetyl-CoA. Okay, this way you have to rule out the options, and this is the important concept. Got to know about the important enzyme pyruvate carboxylase, and if you want to know in detail discussion about gluconeogenesis pathway, link has been given in the description. You have to access the game app where all the biochemistry related videos has been uploaded and recorded by me. So, please go and subscribe the game link and access the game app which has been provided in the below description. Thank you.